Hi, I'm Ms. Trevino, and today we're going to be talking about enzymes. Make sure you did your attendance check for today. And like I said, we're talking about enzymes. You do have an assignment for today, and yes, it is for a grade. Also, make sure to do the lesson check after these notes. It's about body systems, and we're going to be talking about body systems in tomorrow's discussion. Today, we will identify and investigate the role of enzymes. You will be able to describe the role enzymes play and explain how enzymes work to speed up chemical reactions. Let's review everything we talked about last week. Remember that carbon is everywhere in the atmosphere, ocean, fossil fuels, and animals. Carbon is the primary component of organic compounds. Every living organism contains carbon in some way. It's the basic building block to most cells in your body. It can easily bond with other atoms, such as hydrogen, nitrogen, oxygen, and phosphorus, to form biomolecules. A monomer is a simple molecule that has the ability to chemically bond with other molecules in a long chain. Remember that monomer means single part. A polymer is a chain of an unspecified number of monomers. Essentially, monomers are the building blocks of polymers. Polymer means many parts. Biomolecules are large organic molecules found in living things and are the essential molecules to life. These biomolecules include carbohydrates, lipids, proteins, and nucleic acids. All biomolecules can be identified by their structural formulas and are uniquely made to perform specific functions. Carbohydrates are composed of carbon, hydrogen, and oxygen. Carbohydrates are the primary source of quick energy in living things. Plants, some animals, and other organisms use carbohydrates for their structural purposes. Monosaccharides are monomers of carbohydrates. They're referred to as simple sugars. Polysaccharides are large biomolecules formed from complex carbohydrates. Lipids are composed of carbon, hydrogen, and oxygen. Lipids are a large and varied group of biomolecules that are used for the long-term source of energy in the form of lipids called fats. Lipids provide insulation from the environment for plants and animals. Monomers of lipids include glycerol and fatty acids. Remember that the membrane surrounding each cell is also composed of a lipid bilayer. Proteins are made of carbon, hydrogen, oxygen, and nitrogen. Each cell in a living system may contain thousands of different proteins, each with a unique function. They control the rate of reactions, transport substances, fight diseases, function as hormones, and regulate cell processes, called enzymes. Today we're going to focus on enzymes. Proteins can be found in your hair, nails, bone tissue, and muscle tissue. Monomers of proteins include amino acids, and polymers of proteins include polypeptides. And finally, we have nucleic acids. Nucleic acids are made of carbon, hydrogen, oxygen, nitrogen, and phosphorus. Nucleic acids are key macromolecules in the continuity of life. They carry the genetic blueprint of a cell and carry instructions for the functioning of the cell. Monomers of nucleic acids are called nucleotides. Nucleotide monomers then come together to make two main types of polymers: ribonucleic acid, RNA, and deoxyribonucleic acid, DNA. Remember that a nucleotide is composed of three parts: our phosphate group, our 5-carbon sugar, and our nitrogen base. And again, nucleotides come together to make RNA and DNA. Finally, we're on enzymes. Enzymes are proteins that act as biological catalysts. Catalysts speed up the rates of chemical reactions, meaning they make reactions go faster. Enzymes play essential roles in controlling chemical pathways, making materials that cells need, releasing energy, and transferring information. Like I said last week, enzymes are very important. Inside your body, thousands of different chemical reactions are occurring every second. Right this very second, your body is breaking down nutrients. If you ate breakfast or lunch today, your body is breaking that down. It's also building new materials and maintaining your body tissues. Your hair, nails, and ears are constantly growing, and that's because your body is constantly making new cells. All of these critical life processes and many more are driven by chemical reactions. However, many chemical reactions occur too slowly to support life processes. For example, chemical reactions that are absolutely essential in creating the building blocks of DNA would take 78 million years in just water. In our body, since we have different chemicals inside, this reaction happens in 10 milliseconds. So, what would take 78 million years just takes 10 milliseconds in our body. How is this possible? This reaction happens so quickly in our body because of enzymes. These are protein catalysts that speed up the rate of chemical reactions. Enzyme catalyze a reaction by lowering the activation energy, which is the amount of energy needed to begin a chemical reaction. All chemical reactions need activation energy to get started. Activation energy is needed to speed up the motion of molecules to increase the frequency and force with which they collide. Lowering activation energy dramatically affects how quickly a chemical reaction is completed. Enzyme catalyzed reactions occur extremely fast. Many happen millions of times faster than an uncatalyzed reaction. Just think about helping your parents or helping a friend move. If your friend was moving to a one-story house, it's one level, you're able to move boxes easier. Since you're able to move them easier, you're able to get it done faster. Now, say your friend was moving to a two-story house. Now you have to climb a set of stairs to help them move boxes. It's going to go a lot slower because you need a lot more energy to get that done. So, an enzyme would be the first story, very little energy needed. Without an enzyme, it would be moving up to a second story. A lot of energy is needed. This graph demonstrates the effect enzymes can have on a chemical reaction. Notice that the blue line on the graph shows the reaction taking place without an enzyme. The blue line needs a lot more energy to get the same products as the red line. The red line shows the reaction with the enzyme. Notice that the activation energy needed for the reaction with the enzyme is much lower than the reaction without the enzyme. This means that the reaction can occur a lot faster. It is important to note that whether or not reaction has an enzyme, it still makes the same product. Think about using a flash pass at Six Flags. You can use a flash pass so you don't have to wait in line with other people. You basically cut the line and get on the ride faster. The flash pass would be an enzyme, and it lets you skip the wait time in line. But you and a person without the flash pass are still going to ride the same ride, so it still has the same outcome. The only difference is you get to ride the ride faster than a person without the flash pass. So, what do enzymes do? The reactions involving enzymes, the molecules at the beginning of the process, are called substrates. The structure of a substrate is physically and chemically matched to the substrate, just like a lock and key. So it's kind of like a puzzle piece. They fit together. The substrates bind to the site on an enzyme called the active site. Enzymes are specific to a substrate based on the shape of its active site. If the shapes do not match, the substrate and enzyme will not connect and no reactions will occur. They have to match and they have to bind together for the reaction to occur. When the enzyme and the substrate are joined at the active site, this is called the enzyme substrate complex. Once matched, the enzyme converts substrates into different molecules called products. After the enzyme has performed its work, the product or products that it makes drift away. Once the product or products drift away, the enzyme is then free to function in another chemical reaction. It's important to note that enzymes are reusable. They're not used up during a single reaction. They can keep going and going, making products as long as they do not change their shape, which is called denaturing. We mentioned that last week, and we'll talk about that more again in just a few minutes. So, these are some common enzymes. Cells use enzymes to catalyze many reactions inside the body, including reactions involved in growth, reproduction, digestion, and nutrient absorption. Some cells also excrete enzymes outside their cell walls to break down molecules in the environment. For example, E. coli bacteria excrete enzymes to break down food in their environments into molecules small enough to pass through the cell wall and into the cell. Protease is an enzyme that breaks down a long protein into smaller chains called peptides. A protease, also called peptidase, is an enzyme that can break down a long protein into smaller chains. Amylase helps to break down starch, or carbohydrates, into smaller sugar molecules. Amylase is inside your saliva and also your small intestine. Cellulase breaks cellulose molecules down into simple sugars. Bacteria in the guts of cows and termites excrete cellulase, and that's the reason why cows and termites are able to digest things like grass and wood. Maltase helps to break down maltose and sugar. DNA polymerase helps build up DNA polymers. Protease breaks down protein, and lipase breaks down lipids. Hopefully, you've noticed what all of these enzymes are common. With a few exceptions, the names of enzymes are all going to end in ASE. The name depends on their processes and the substrates they interact with. So, anytime you see a word ending in ASE, you should note that that's an enzyme protein. Remember last week we talked about words ending in OSE? That stands for sugar, which are carbohydrates. ASE is enzymes, OSE is sugar. Enzymes catalyze a reaction by reducing the activation energy needed for a reaction to occur. However, enzymes need to be tightly regulated to ensure that levels of the product do not rise to undesired levels. They want to make sure that they're not making too many products. This is controlled by enzyme inhibition. Inhibitors are chemicals which bind to an enzyme to stop its activity. This binding can be permanent or short lived. Inhibitors can bind directly, blocking the active site, or by binding somewhere else on the enzyme that causes it to change its shape of its active site. If the active site is blocked, no substrates can bind to the enzyme. If an inhibitor changes the shape of the active site, then the substrate that it would normally match with can no longer match with that enzyme. Because enzymes are catalysts for reactions, enzymes can be affected by any variable that influences a chemical reaction. If the protein is subject to change in temperature, pH, or exposure to chemicals, the activity of enzymes can be affected. Many enzymes are affected by changes in temperature. Normally, enzymes produced by the human cells work best at temperatures close to 37 degrees Celsius, the normal temperature of the human body. Imagine getting a fever. Your body turns hot. Your body temperature rises. Because of that, your enzymes are denatured. Your enzymes are changed. That's why whenever you are sick with the fever, your body isn't working like it normally does, and you often feel drained and tired. Enzymes work best at certain ionic conditions and pH values. The pH scale is a scale that is used to rank the relative basicity or acidity of substances to other substances, based on the amount of hydrogen ions. The scale is logarithmic in nature. The pH scale is based around pH 7, which is neutral and represents substances which are neither an acid nor a base. It's usually water. For example, the stomach enzyme pepsin, which begins protein digestion, works best under acidic conditions, but it has to be the right amount of acid. Imagine eating way too many hot Cheetos. Your body starts to feel uncomfortable and you start burping a lot. You don't really feel like you're digesting the food. Well, that's because you raised your acid level in your stomach, so the enzymes aren't working like they normally should. An enzyme losing its shape is what is known as denature, like we discussed last week. Denaturation is often reversible because the primary structure is preserved if the denaturing agent is removed, allowing the protein to resume its function. But sometimes denaturation is irreversible, leading to a loss of function. Remember the boiled egg scenario. When an egg is fried or boiled, the liquid egg white is denatured when placed in a hot pan, changing it from a clear substance to an opaque white substance. Not all proteins are denatured at high temperatures. For instance, bacteria that survive in hot springs have proteins that are adapted to function at those temperatures. There is an Amoeba Sisters video over enzymes. Be sure to watch that video because it talks about a lot of important concepts. You also have your assignment for enzymes, and yes, it is for a grade. Please let me know if you have any questions. Thank you and goodbye.